From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on September 12th, 2025. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 108th episode of Matters Microbial. Many thanks to every listener, viewer, guest, and supporter. Keep those likes, listens, and views coming. I learn something when I do each podcast, and I hope you do as well. First, a very sad note and a remembrance. My friend of 30 years, microbiologist Dr. Kimberly Walker, passed away suddenly last weekend. She was a remarkable person and was a guest of this podcast in episode 44. My wife, Dr. Jennifer Quinn, created this Lux art of Kim. I will post a link to her well-written and very moving obituary. I will miss Kim very much. My microbiology course and laboratory are off to a great start because of the wonderful students I'll miss when I'm no longer teaching at this institution. Perhaps I can find a way to still do microbial outreach and become a microbial evangelist after retirement. I hope so. As I mentioned last week, I have students swab or streak the inner walls of their water bottles. As you can see here, there exists quite a diversity of life there, different colors, textures, and sizes. By the way, I'm using R2A medium, which is a type of medium designed for analyzing water contamination. So it's a perfect source. In any way, I want to emphasize that these are completely normal inhabitants, since after all, it's a microbial world in which we all move. Micronaut Claire is getting used to the idea of her water bottle buddies, as seen in this short movie. Our next goal is for each micronaut to isolate 3 candidates from their water bottles for further study. Remember this original possible siderophore-producing swab seen here via fluorescence? Well, that micronaut has restreaked one of those fluorescent colonies, and it looks pretty good for isolation, seen here. It was also quite a bit of fun to have my micronauts hone their dilution and plating skills with E. coli containing plasmids that in turn encode pigment genes, as seen in these flasks. These plasmids were developed by Sebastian Kosiba, a former guest of this podcast, episode 9. When I spin down the cell pellets, seen here, the colors are quite lovely. One of them, of course, is our good friend GFP, green fluorescence protein, seen here under UV light, absolutely blazing. As always, I remain interested in Lux art. Using large petri dishes with nutrient medium and a very bright photobacterium strain, the students truly impress me. While my wife, Dr. Jennifer Quinn, is a great Lux artist, as seen in this portrait of the actor Jeff Goldblum, my Bio 350 students amazed me as always. Here is a good Lux art piece of a lighthouse. Even a bit of art history humor, as seen with this Lux art, with a hat tip to Magritte's "This Is Not a Pipe." Since I love astronomy, this Lux art of galaxies seen here was lovely to see. And look at the intricate and lovely great Lux art here. Such talent. I truly enjoy putting on Lux art workshops and seeing the amazing creativity. I hope to continue to do this in the future. I often tell students that we don't know enough about the deep past and that some scientists have tried to look at ancient DNA in a genuine fashion, unlike the Jurassic Park movies. But how to do this, and what can we learn by doing so? Thus, I am so very happy to welcome Dr. Laura Weyrich of Pennsylvania State University's Department of Anthropology to discuss her research group's work studying the paleo microbiome. I love that word. Welcome to the Quality Quorum today, Laura. It's wonderful to meet you, even virtually.
Thanks for having me, Mark. It's a pleasure.
This is a subject of a big interest to me, and I also note that you wear many hats. Not like these, but in terms of where you are, what you're doing. Would you care to give us a thumbnail sketch of those things first so we know you?
Sure, no problem. I'm an associate professor of anthropology and of bioethics, so two different departments here at Penn State. And I'm also a co-hire with the Huck Institute of Life Sciences here. I have appointments also at the University of Adelaide in the School of Biological Sciences. So I have lots of different hats as both a microbiologist, an anthropologist, and an ethicist.
And you also collect frequent flyer miles.
I do, quite a few of them.
All right, so I love the idea of the paleo microbiome. And one of the things that when we talk about microbiome research on this program, we'll often have a situation where people will say things along the lines of, how do I know I've preserved everything that's there? For example, when we were to say swab a water bottle and send those off for sequencing, has that DNA broken down? Have those cells survived? Those kinds of questions. How do we look at the deep past then?
Most of us use DNA because DNA does have, a long half-life. It's, surviving in ancient individuals and in the sediment and, in other places for quite a long period. You can also use things like ancient proteins, ancient lipids, even some studies on some ancient RNA, although those are just emerging at this stage. But mostly we use DNA. DNA is kind of the foundation of a lot of our work.
I think it's really interesting because, and I want to be very clear about this, because there are all kinds of people that join us to listen or watch what we do. Sometimes the media makes things look a little bit different than the reality. For example, the idea of creating extinct animals in this fashion. And I'm sure you have some things to say about that.
Yes, exactly. So the field of ancient DNA has a crazy long history. And there's been a lot of poor science done in the field, but also a lot of really remarkable, amazing science done in the field. And so, yeah, it's amazing to see what a journalist will run with sometimes. It's also amazing to see what sometimes non-scientists or people who are not trained well in ancient DNA scientists will also take and run simply because, the odds of getting a really high-profile paper can be in your favor when you're talking about ancient DNA.
One thing that I want listeners and viewers to know, and when I say what I'm going to say, I'm not casting any judgment or aspersions on anyone. But for me, when the kind of research that I've always done as an old school microbial geneticist, controls were royalty. You had to have good controls. And I know that when people were first doing microbiome work, they weren't doing no sample controls. And by that, what I mean is the small amounts of microbial DNA that were even in the reagents that were being used. So they weren't necessarily looking at the target. And for this reason, some types of work where you're looking at low availability, we have to be exceedingly cautious. And I hope that's clear to everyone.
Yeah, that is one of the biggest things that we struggled with early on. And it actually hampered the field. I would say, ancient microbiome research didn't happen for probably a good decade because contaminant levels were absolutely just too high in reagents. And it took companies like Mobio cleaning up reagents that were available for use in order for people to actually be able to see these really, background, very tiny levels of endogenous or true DNA that corresponded to ancient folks. And my lab has done a lot of work on this in both ancient spaces as well as modern spaces because we work a lot with Indigenous communities and other communities on modern oral health issues. And, focusing on both ancient communities, but also the oral microbiome means that we have a double whammy of concerns when it comes to contamination. As I'm talking to you now today, I'm spraying oral microbes everywhere, right? And so if you are going to get a sample and, reconstruct an oral microbiome, you have to be concerned about the contamination that could have been present when it was collected and the environment in which you're collecting it, all of that. And so taking lots of controls, during your sampling, as well as, during your extractions, during your library preps, during your sequencing, all of the controls you can probably think of. And in fact, sometimes maybe we do too many controls, but, better to know than to not know, and, have your results be a mess later. And we also do that in an ancient DNA context, right? So we're swabbing museums where, individuals may be curated or housed. We're swabbing, anything in the field site, if we have access to that. We're taking sediment samples next to individuals that are unearthed, out of the ground. We're also swabbing our labs on a regular basis, right? We're including those extraction controls. We're including those other controls. We've even done inside the tooth controls. And so, the very first paper we published on this in 2013, we had a whole panel of controls to really, I think, get over that burden of proof and show that there are indeed these oral microbiome signatures in ancient individuals that we can reconstruct if we are mindful of contamination, and if we are mindful of all these other sources that can provide oral DNA, into our samples. Does that make sense, Mark?
It does. Absolutely. And even the kinds of stuff that I do, I have a colleague and we were doing some work on cloacal microbiomes in lizards. Don't get me started.
Awesome.
That colleague of mine got so paranoid, and it was because of me, that she would remove a swab and wave it in the air, and that was one of her controls.
Yeah. We do things like set tubes out, on the bench
Yeah.
when we're working all day and just see what's in those tubes. And you would be amazed, especially if you're using techniques that can pick up those sort of low biomass signatures. You'd be amazed how much DNA ends up in that tube.
And I want to say, and this sounds like it isn't involved, and I'm not going to put you on the spot about this, but I'm an entropically challenged individual of great age now. And I well remember, and I won't name the person or anything, there was this big to-do about isolating ancient yeasts from amber. And as I read this, and I'm not making a judgment, I was thinking to myself, how do you know that's where it came from, that sort of thing? And I want listeners and viewers to remember, when you're doing this kind of work, what could go wrong. And so much of the time that we try and put things out in the media as quick as we can with like these great titles, now we're eating more beets, that kind of thing. And science like this is very serious.
Yeah, it's like I said before, it started out on a really rocky front, this field did. And, in the early 90s and 2000s, there were a ton of papers that came out isolating bacteria and yeasts from amber. There's been a ton of research since then where they actually employed controls and they employed all the standards that we use in ancient DNA to authenticate the fact that this DNA might be ancient. And they were not able to replicate any of those sort of studies. And so that tells you something, pretty strongly about some of those early studies. They were done in labs that were not cleaned, not set up for this type of work, not set up for ancient molecular work. Even the organisms that they were able to, obtain or culture from these, they never went through and did genomic sequencing and sort of placed them in phylogenetic space to show they fell, ancestral to modern strains. They were not able to authenticate the damage or anything else that, should have been in the dead, neighboring potential microbes in some of that amber. So there were just whole bunches of issues because people weren't trying to show their results, beyond a shadow of a doubt, right? It gets so exciting, and we are excited by the science. It's really exciting. It's really amazing. But at the same time, you have to be kind of your worst critic, right? and evaluate your stuff, with the fine-tooth comb. So that was really early on. And even when I teach lectures here at Penn State about ancient DNA and the history of ancient DNA, we use a lot of those papers as examples to say, okay, what could they have done differently? What could you have included as controls or authentication criteria, to really teach students that I think every paper you read is wrong in some way, shape, form, or another, right? We don't always get everything correct.
Right.
And we're learning constantly as scientists, right? Things change over time. But B, that, you have to be really critical of your work, and you have to think about, okay, what are all the things that we can do, right? To try to build evidence to show that we have a real signature here.
Oh, I like the way you put that so well. I had a friend when I was a postdoctoral scientist, postdoctoral student. I guess we're not supposed to say it. When I was a postdoc, down the hall, who was studying relationships of paternity in gorilla populations. And this is in the relatively early days of PCR. And he would keep amplifying his own DNA. That's not a joke, because we're pretty closely related. and some skin flakes, and they would react with the primers. So he had to really beat himself up with controls. And it's a really good idea.
Yeah, absolutely. I mean, it's pretty well known in our field that even some of the first papers, I think the first paper by Svante Pabo on mummies in the 80s, it was shown later to be some of his own DNA, right? And so, somebody who won a Nobel Prize for ancient DNA, right, got the first paper wrong. So, you have to have those controls, and it was a great lesson, right, for everybody to learn and to figure out how to do these things and how to do them in ways that are reproducible and rigorous.
And without taking us off topic at all, I think it's important for listeners and viewers because I have deep concerns about scientific literacy to the public. I'm not putting anyone down, but so many things are thrown at you with headlines. So to understand that science is a process, and when done correctly, it's self-correcting because other people are trying to replicate your work and extend it. There's nothing more tragic than a beautiful observation that you can't replicate. But I got to tell you, it happens to everybody.
Yep.
Right? It's cruel beyond words, right? But if it doesn't happen repeatedly in someone else's hands, it's not real.
Exactly. And even if things aren't happening in the same way across labs, that difference may be something really interesting and really worth chasing and telling you something about what you're doing and how you're doing. And so even just paying attention to those differences, they're important to chase down and to almost celebrate in a way because they're telling you something interesting about the science that you're doing instead of try to, sweep them under a rug or hide from them. That's one of the lessons we try to teach most of the students here on a regular basis in one of the ethics classes I teach.
Oh, and it's great that you're doing that as well, because many people I talk to outside academia don't appreciate the importance of ethics in the work that we do. Mm-hmm. And certainly people understand that when you talk about genetic counseling, for example, but this is relevant here. So your own system that you've been looking at that we'll discuss a little bit today has been to look at ancient oral microbiomes from
Neanderthals and ancient humans. Yeah. Yeah.
And you have lovely photos too. So I hope we can work them in.
Yeah, sounds great. Yeah, my team has really been involved in trying to push the envelope in the context of ethical approaches to this science, and really thinking about how we recognize and celebrate Indigenous histories and Indigenous rights in the context of our work. So I have a hat that's not just a scientist role, right? I also get to put on an ethicist role. And I think it just enriches our work so much more as a scientist to think about the ethics of how we're doing it and to integrate communities and get that community collaborative approach rolling with all of our projects. So I've been really fortunate to work with many different communities, many different collaborators from all around the world in that space. And we've been trying to write, now at this stage, ethics papers on how we do not only ancient human DNA, but how we do ancient human microbiome DNA. Because as you know, Mark, microbes can say a lot about a person, right? They can be identifiable. They can tell you about what diseases people had. There's a lot that can influence our interpretation and our understanding of people and past populations. So, having input when we're doing microbiome research is also really, really important. We can't just do that because it's microbes, not humans. No, these microbes are coming from humans and they say a lot about humans. So we've got to care about the ethics of how we go about doing our work.
Oh, absolutely. The number of times that I've had to explain to students that when you collect microbes that might have biotechnological applications, hello, Yellowstone, there has to be a certain degree of responsibility to the people who were in the area before you were.
Yeah, absolutely. And it's even, I think, more important in the microbiome field as we move towards technologies. And I have a small company, a small biotech company called Microversal LLC, and we are leveraging microbes from people and transplanting them into other folks to improve their health. If you're obtaining those from Indigenous people, those Indigenous people need to have a say in how that's being done. And I think also have rights and also need to have monetary compensation, right, for the microbes that they've curated for decades or for millennia, right, and that you're now using and employing in sort of biotechnology operations. So I think for the microbiome field, broadly speaking, we need to sit down and have a think about how we treat microbes and how we think about them. But yeah, that's a whole nother podcast, probably, Mark.
Oh, we've had several podcasts where we've talked about issues related to this, and sometimes people will come at me if you don't mind my saying so, that sounds like I'm trying to talk about sociology and all the rest. And I'll say, it is part of the whole planet. And we don't have a right to everything that we find. We have to be cautious about that, as we would of some material possession that someone had. It's really no different as far as I'm concerned. And I'm not saying don't do the work and all the rest, but I think we're on the same page, and there's been some wonderful work, and I am so delighted that you take the attitude that you have on this. And I'll put some links in the show notes on that. Incidentally, for listeners and viewers, I will certainly put a link to the quite wonderful website that you have, which I think you should have a big stack of hats as an image for it, because it's really true when you look at the kinds of things taking place.
Yeah, we are very interdisciplinary, if not anything. It's one of the reasons I came back to Penn State after finishing a PhD here many years ago. But we need that interdisciplinary support, and we need to be in a place that champions, the ability to integrate all these different lines of evidence into the science that we do.
So can you walk us through how you would isolate some, I'm going to call it paleo DNA for lack of a better term?
Yeah, that's great. We just call it ancient DNA colloquially in the field, but it doesn't really make sense because sometimes we're working on people who lived in sort of more historic periods, but we still call it ancient DNA. So, it's kind of these methods that we use that are ancient DNA methodologies. So then we just generally call things ancient DNA. But for us, the primary sample or tissue type that we are after when we're trying to reconstruct ancient microbiomes is a substrate or a sample called dental calculus, which is just calcified dental plaque. Dental plaque is that bacterial biofilm that grows on your teeth, especially after drinking something sweet. You can feel that sort of slime on your teeth. That is that bacterial biofilm growing and reproducing in real time. And if you, go to sleep at night with that plaque still on your teeth, you will get that plaque will form into a rock hard like matrix, which is what we call calculus. Has nothing to do with math. It has to do with more of the sort of mineral structure. But it is this sort of cement-like matrix with the microbes encased, in this rock hard type substrate. And so it mineralizes at night when you go to sleep and the pH in your mouth changes. And that means that it locks those microbes in place while a person is still living. And so, when you go to the dentist and you're having that sonicator in your mouth or drills or whatever, where you're not having cavities replaced, or if you're having scaling, right, where you've had your some things removed out of your mouth, that is removing that dental calculus from your teeth. So a lot of people in industrialized societies today who have access to, modern dentistry, we don't build big amounts of dental calculus. But in the past, prior to modern dentistry, people would build very large pieces of calculus in their mouths, right? Some of them are larger than the teeth themselves, right? The calculus would get really, really big over time and space. Like you can imagine your cheeks being bulged out in some of the people that we've seen. But this is this wonderful record in the archaeological record that we can go back in time and space and reconstruct types of oral microbes that lived in people's mouths. Occasionally, we also get respiratory microbes or, things that might be coming back up as well through one way, shape, form, or the other from the gastrointestinal tract, or maybe even from the lungs. But it's predominantly these bacteria that form this biofilm on the teeth. So most of them are what we would consider sort of dental plaque microbes. And, for those of you that don't know, your mouth has a ton of micro environments, right? You have different microbes in your saliva versus on your tongue versus on your teeth. And even with on the teeth, you have different microbes in the front of your mouth compared to the back of your mouth, compared to the side near your tongue or the side near your cheek, right? So it's very involved. But these ones that sort of stick to your tooth surface, those are the ones that we spend a lot of time looking at. And so there's other samples you can use like coprolites, which are preserved feces. We can now use sedimentary ancient DNA to pull out some microbes. There's all sorts of other things. You can, scrape ceramics and, things for making cheese to get some of those ancient fermentation microbes, right? But the bulk of our work, because we're interested in the microbiome and we're interested in its association to both infectious but also chronic disease over long periods of time, we really focus in on dental calculus. So that was a rambly answer, but that gives you the sort of set the scene for the sample type that we're using. And I believe we've got a picture of that you can pop up and show that sort of biofilm that's, grown on the gum of a person who lived in medieval England.
You showed a really lovely image of two different jawbones, and I wonder if you could walk us through or tooth us through the differences and similarities between them?
Yeah, no problem, Mark. So the top one is actually from a community of folks that were hunting and gathering most of their food in Japan. And this would have been somewhere between 5 and 3,000 years ago. And we can see again that they have great oral health, right? That we don't see, huge amounts of decay. We don't see gum disease, right? Their oral health looks really, really great. And we had the privilege of reconstructing the bacteria that lived on their teeth in a paper that we published several years ago. And we were able to compare that to people who lived in more recent periods who did a lot of farming in Japan. And this is true not only in Japan, but it's true elsewhere in the world, right? This is true in Europe, all over the place. We've also done some active research to show that it's true in other continents, and that's research that'll be coming out here shortly. But we see this large change in oral health across the invention of agriculture. Basically, we start eating a lot more carbohydrate in the diet, and we see an adaptation of microbes. And we can do this by looking at Streptococcus species and their individual genomes and seeing that they acquire a lot of genes that are required for disease in the mouth. So really, the advent of agriculture is the first major shift away from a core hominid microbiome to microbes that are much more likely to cause disease in the mouth, or a microbial community that's much more likely to cause disease in the mouth. We then see additional subsequent changes sometime, we think, during the Industrial Revolution or rightly thereafter, and we have a paper coming out on that here shortly. So there are changes associated then again with the Industrial Revolution, but we kind of maintain that agriculture microbiome, at least in European communities, for probably about 6,000 years. And that just breeds, a lot more microbes that can also cause disease, or microbes that do well in disease states, right? So by the time we get to the Industrial Revolution and we see another change in the microbiome, it doesn't mean better health things. It means even worse health outcomes. And we know that there's been another subsequent change since the Industrial Revolution. We actually think this is what we call the Great Acceleration change. And this Great Acceleration change is probably the industrial signature that, folks are seeing today. The gut papers all say this industrial versus non-industrial microbiomes. It's probably a great acceleration or a not great acceleration microbiome. And this is probably something that happened post-World War II with, rapid changes in, say, for example, access to antibiotics, preservatives in foods, oral hygiene practices like toothbrushing. So we know there's been several different changes, but that first big change was really when we went from hunting and gathering most of our food to agriculture and really changing our diet drastically, which also shifted the microbes in our bodies.
Could you possibly run through the changes that you've seen between, for example, the medieval oral microbiome and the modern ones? Are there specific changes that you can address, or is it there's too much noise? I can't tell.
Yeah, that's a great question. So we've spent a lot of time looking at this specifically in England, and we did that because it's the birthplace of a lot of industrial type practices, like a lot of the burning of coal and the mechanization that's associated with the Industrial Revolution occurred first in the United Kingdom, in England. And so we see this shift from the medieval period into the industrial type era. And we're still investigating why that might be. We think it might be due to sort of heavy metals or pollutants that are released in large quantities in the environment. But we also think it might be related to just an increase in the amount of sugar that people consume. And on top of that, the sort of, what we would consider modern types of sugar, the concentrated, white powders, right, of sugar that we now have in our daily lives, right? That's what starts to change during the Industrial Revolution. And so we see this sort of increase in different genes that are required for carbohydrate digestion, right? And in the oral cavity, we also see this increase in microbes that do well under those sort of simple sugar type environments, or do well in those environments. And so they acquire genes to, for example, fight off other microbes or other microbes that do well in those simple sugar environments. So we see, both whole community changes during that industrial era, but also individual genome changes in specific species that are linked to, this very specific adaptation to a new industrialized type lifestyle. And then, as I mentioned before, there is this change again that happens during the Great Acceleration. And we're still trying to get a handle on that. It's actually much more difficult ethically to study that time period because the people who existed from that time period are usually your grandmother, or somebody, that wouldn't be, curated in a museum, for example. Although we have worked with places in both France and Australia with populations of people who donated their bodies to science in the 50s and 60s, where we're able to clean their calculus and reconstruct microbes that lived in people across that barrier. But those are kind of, again, the more recent changes that have happened since the medieval period, when we had pretty horrible oral health until today. And we're able to kind of curb that oral health through toothbrushing and trying to get rid of the microbes in our mouth all the time.
Could you clarify for us a little bit? Because I know listeners and viewers that are not savants in microbiology will be interested in this, because we're all taught about toothbrushing. And I'm going to call that the idea of cleaning things as opposed to other practices with our teeth. In other words, are we removing things that should be on our teeth when we overbrush?
Yeah, that's a great question. And a lot of times when I'll give public seminars, I'll have people come up to me and say, well, I should stop brushing my teeth then and let these microbes, be in my mouth and support my health. The problem with it is if you have ancestors that went through the Industrial Revolution or went through the Great Acceleration, you probably have what I would consider a disease state in your mouth today. Like, you have effectively kind of a bad microbial community. It's highly adapted to sugar. We know that it's linked to things like caries and periodontal disease. So brushing does remove those microbes and it keeps them at bay, right? It really sort of, tries to kill them and tries to remove them. Everything in your toothpaste are highly antimicrobial, right? Toothbrushing is physically removing them. Maybe you floss and physically remove as well. Maybe use mouthwash and with some ethanol, right? There's, alcohols that kill those microbes in your mouth, right? So you're doing all these things to literally kill off the microbes in our mouth because you have the wrong types of microbes from an evolutionary perspective. It's really an evolutionary mismatch from what you probably should have. So what we are trying to do is reassemble those communities from the past that you maybe should have that aren't associated with disease and put those back into your mouth. So if you were lucky enough to get an oral microbiome transplant, then don't brush your teeth and keep those microbes around, right? Versus if you are still, somebody who has those industrial microbes, brush your teeth every night, brush them twice a day, because you have to remove those or you're going to get these diseases associated with those bacteria.
There's been a lot of talk, Laura, about periodontal disease being associated with other types of health challenges. I've heard about a relationship between porphyromonas and gingivalis and heart disease, for example. Is there anything that you found in your research that's relevant to this concern?
Absolutely. This is the primary thing that we are trying to examine when we work with Indigenous communities and we look at oral microbiomes. A lot of this is linked to things like periodontal disease. So right now we're running a very large intervention trial with the Australian Centre for Population Oral Health in Australia, in Adelaide, with Lisa Jameson, who's the director there. And we're really trying to understand how getting oral health care and, really cleaning the teeth and doing all of the preventative work that you might need to might not just improve oral health, but might also improve systemic health markers. So things like gum disease or periodontal disease that's a risk factor for developing many other chronic diseases. So things like gastrointestinal cancers, like colon cancer, things like Alzheimer's, heart disease, diabetes, and rheumatoid arthritis. So, those are the diseases that we've studied that we know of so far that are linked to this, right? So improving your oral health should have knock-on effects elsewhere in your body, and that's what we're actively investigating. But it also, on the flip side, means that if we're going to transplant microbes, we better get them right, because what you don't want to do is transplant microbes that then have knock-on effects for other chronic diseases, right? And put you at risk for other diseases. So it's tricky to figure out how to do this correctly. But at the same time, it's really important that we're thinking about oral health, not in the context of your teeth and your mouth, but in the context of your whole body. Just like it's important to think about the gut microbiome, as a systemic or as a whole health type treatment and not just, something to fight a gut infection. I don't know if that answers the question, but I hope, thinking broader about, again, not that reductionist approach of just what's happening in the mouth, but how it leverages whole health in the body or how it might influence other systemic diseases, I think is really, really important in our work.
You've mentioned that you are involved with an attempt to do oral microbiome transplants. And I was kind of curious as I thought about that, are any members that, because when you're doing this, I don't know if it's like a group of microbes that you take or a select number. I don't know enough about it, but are any of them related to the things that we've talked about in older human microbiomes taken from folks that didn't seem to have, quote, bad teeth, end quote?
Yeah, that's a great question. So we are developing oral microbiome transplants with Microversal LLC, and I guess just as a conflict of interest, I am the CEO of the company, so I, I have to put that out there. And what we're doing is transplanting, depending on the formulation, somewhere between 150 and 230 microbes into each person who receives them. So it's a whole entire community that we are transplanting. Some of the key things that we're looking for are species identified in the archaeological record as microbes that can outcompete known pathogens today. So, for example, people in the past had very different species of Streptococcus in their mouth, some of which, again, we don't see in industrialized populations today. And some of those species we know can outcompete things like Streptococcus mutans, which underpins cavities. And so, if we can enrich for those types of microbes, we might be able to really, use an ecological approach to limit the outgrowth of pathogens that are associated with disease. So that's the type of, ecological framework that we're using to try to employ here when we're thinking about leveraging ancient DNA into modern folks. We are not trying to culture microbes, from Neanderthals. That's not possible. I wish it was, but it's not. We have to use microbes that are from living people today, but from diverse communities, from people all over the world that may still have some of these unique non-industrialized microbes, right? That we might be able to bring into the clinic and to leverage in ways that we wouldn't by just studying people who all come from industrialized areas.
One of the advantages to your work, it's very obvious to you, but I want people to know this. If you have a sample where you can preserve some DNA from microbes, how do you know it's representative, that what you get is representative? But the good news is we know a fair amount about the dental microbiome. And so that gives you kind of an idea that, yeah, we're in the right neighborhood. And that would be very helpful, because if you didn't have any kind of background, how would you know if it was representative?
Yeah, that's exactly it. When we get, a sample from an ancient individual who's passed away, we do have to bring it into the lab. And so we have a very specialized laboratory facility. It's effectively a BSL-3 facility, but we run everything in reverse, because instead of trying to protect the technician from the sample, we're trying to protect the sample from the technician. But it is, still full gown. It's all HEPA filtered air and UV treated, and it's an incredibly sterile environment. But not completely sterile, and that's important, right, for contaminant concerns. But what we do is we decontaminate calculus first. So we actually take our samples and soak them in bleach. And, in a modern experiment, you wouldn't do that. That would be, insanity, right? But we soak our samples in bleach. We also UV treat them on the outside. So we blast them with UV. And the idea there is that we're trying to remove any DNA from any microbe on the outside that could be tied to an environment. We then wash the sample, and we've done experiments where we've taken that wash to even look at the types of microbes that might still be present on the outside. And we've also then compared this, right, to like soil from where that person was taken from, the inside of the tooth from where that person was, or from that person that was there, right? So we've done a whole bunch of controls. So we can take, the microbes that are locked within that decontaminated calculus, compare it to all these controls and say, okay, this is what's left. And of what's left, do we think that it's oral? Is it something that we're seeing in people who live today or not? One of the key issues here, though, is that people who lived in the past probably don't have the same microbes, all of the same microbes as people who live today. And, we've seen a lot of that through Justin Sonnenberg's work, right, with these vanishing taxa in the gut. The same thing happens in the mouth. We have, microbes that we know are really high prevalence in ancient people, and we rarely ever see them in people today. So some of this took a while to figure out, and it was through sequencing DNA from many individuals and reconstructing microbiomes across hundreds of people, where we could be much more sure about some of these ancient oral microbes that are now missing in modern populations that are from industrialized areas. So it's a lot of a lot of double checking, a lot of fact checking, a lot of cleaning. But I love, Mark, that you're sort of thinking about how do we know that these are real? Even when we get DNA from microbes too, we have a bunch of criteria we can employ, like looking for fragmentation of the DNA, damage. So some of the nucleotides are actually damaged and they come up as, different nucleotides like uracils. So we can actually look for those sort of signatures as well and make sure the microbes follow a bunch of other criteria that I mentioned, some of those earlier. So it's a long process to be very sure that we're getting true, indeed, signatures from what we're getting. And I think some people in the field are a bit scared as well about some of these microbes that are missing or not. And so they like just focusing on the core things that we know are still present in people today. And that's great, but we're not learning more about the total microbial diversity that's ever been in the human mouth. And we're not also learning how that might change over space and time as people were in different environments, had very different diets, et cetera.
No, this reminds me very much of some of the work with gut microbiome studies and I'm not going to name particular indigenous groups, but non-industrial societies versus industrial societies. And there you have ethics again, of course. But then you have to actually do this longitudinal study to figure out if they really are that way or if it really does reflect how the diet changes over time. And it's not really a surprise because, we're swimming in a sea of microbes at all times. And like the Frank Sinatra thing, it's a microbial world. We just live in it. And I love how you have locked in the dental calculus. You've locked in the microbes in a way that their DNA can be recovered, because I wouldn't have thought of that. I would have wondered about things in bone, and of course, tooth is bone, but you're talking about plaque, which is different.
Absolutely. And people do a lot of work with microbes that are in bone and look at infectious diseases predominantly. So things like Yersinia pestis, there's all sorts of other viruses. There's been salmonella papers, all sorts of things, right, that have come out of ancient, paleo microbiology studies from bone. For example, like inside the tooth root is a great place to, access pathogens that would have been circulating in your bloodstream. So that's really cool. We just don't pick them up very often in calculus. So it really is this different, signal. It's a different signature, and it allows us to access these beneficial, or at least, co-occurring microbes. I guess not all of them are necessarily beneficial, but, these ones that are really truly the microbiome, and not necessarily just the infectious pathogens that, other teams might be interested in.
I love this concept of a core, I'm going to call it a calculus microbiome, for lack of a better term, a core one versus determining if there are groups that are missing. And the reason this appeals to me is there's so much sociobiology that takes place in the oral microbiota. Mm-hmm. I mean, they're talking all the time. So it would be very interesting to me to look for representatives that do that kind of chatter. And there are lots of well-known and well-studied examples today. Be curious to see if those same ones were present then.
Absolutely. And many of those same ones are. I think critically, there are some microbes that are really great, including many of the Streptococcus species, for example, that are really key about binding the tooth enamel, binding the pellicle, which helps you attach physically to the tooth enamel. And you just don't get calculus unless you have some of those species, right? So those are kind of what we'd expect to be core. They have to be there in order to get calculus. And a lot of work has been done studying Streptococcus species, broadly speaking, because they some of the species within that genus underpin things like dental decay or cavities. So some of these absolutely are there. And then there's other ones that are totally surprising. So in a paper we published on the ancient British oral microbiome over the last 2,200 years, we really saw that there were entire microbial ecologies that are largely absent in many of the populations living today. We have a paper that's been submitted that shows that it is still present in some populations in the world, which we're really excited about coming out here shortly. But it is, really surprising that these totally different ecologies, right, with totally different networks, different signaling, that we know those microbes work together in other systems, right? But it's really surprising to see them in the mouth because we don't see them in the mouth of industrialized people today. So there have been some really big surprises, but studying a lot of these microbial networks and the interactions between microbes, which we've seen in other systems, can help us verify that, no, we do have a true community here that's working together. They're talking to each other. They're feeding together, right? And it helps us kind of, again, authenticate them and verify that what we're seeing is real. Wonderful.
Wow. Okay, so we've talked a little bit about looking at medieval British. We've talked a lot about ancient British. And you've also looked, as you mentioned, at Neanderthal tooth calculus. And do we still have the same kind of memberships that we would expect there in the latter?
Yeah, it's a great question. So we do share many of those microbes with Neanderthals. And in fact, one of the most surprising things for us when we sequenced the Neanderthal oral microbiome for the first time was that we had evidence of microbes being swapped or shared somehow between humans and Neanderthals. So, we do absolutely share some of these microbes with, with Neanderthals, or we did at some stage, right? There are some, though, that we think that there are specific maybe Neanderthal strains that probably have gone extinct when Neanderthals may have gone extinct. There are some representations where, they're not microbes that we necessarily find today in people's mouths. We're still trying to do work to try to figure out, is that indeed an oral microbe? Is it contaminant? What's this, where is this at, right? And we're also doing a lot of work to try to reconstruct other Paleolithic and Mesolithic type microbiomes to understand whether or not we can see differences in signatures between Neanderthal groups. There's very few Neanderthals with dental calculus in the world. They're incredibly precious specimens. You're lucky to find a Neanderthal first, but then you're even luckier if you find a Neanderthal that has calculus on it, because Neanderthals had amazing oral health. They had great teeth. I tell people I wish I had teeth as good as a Neanderthal, because they truly did. They had amazing teeth. And so, we're really lucky to even get calculus off a couple of those. So when we do find them, often we can be kind of limited in scope because they're so precious that we'll get, one itty bitty sample to kind of reconstruct what's there. But we do know that at least, the majority of species, the core ones, are things that we probably share across a hominid tree, right? There is this kind of core part of our microbiome with other apes in the mouth. But then there are these other unique species, which, the verdict's still out, and we're doing a lot of active research on right now to figure out, where did they come from? Why are they there? What happened to them as Neanderthals left the planet?
And just as the gut microbiome can differ with diet, I'm certain the oral microbiome follows that same kind of pattern as well.
That's not an easy question. We have done a ton of work to show that the diet influences the oral microbiome in people, and we've traced that over time and space. And now there's been other teams besides ours who have also seen that when we've done very large dietary changes in the human diet, that we do see shifts in the oral microbiome. But the dogma 20 years ago in dentistry was the microbiome feeds off your saliva. Therefore, we don't see changes in the oral microbiome according to diet. That was the dogma. There may have been some sort of changes according to how much sugar you consumed with Streptococcus species, but that would just shift the abundance, not necessarily the composition. So we've really had to fight tooth and nail to demonstrate that the oral microbiome does shift according to dietary change.
Did you just say tooth and nail?
Yeah, I did. Thanks for catching it.
Yeah, so we'll take a deeper bite into that when we're doing it, right? It's going to go on. We'll scrape to the bottom. Oh, it's going to get bad.
You can keep going on and on. It's great.
Yeah, I'm sorry.
I love it. But
no, and I really love this in general because, and I want to say this very carefully, when I talk about microbiome work with non-microbiome people, they tend to be very expansive at what you can find out. But when I talk to people who do the work, they're very cautious. And that makes me happy because so much stuff could be oversold. By the way, I wanted to tell you one joke that I tell my students a lot. We've all heard the expression a sob story, right? So there have been a number of papers where someone will swab an area and say, yes, there are microbes because it's a microbial planet. So I call those swab stories. So I think now you have swab stories in your clean room.
Yeah, I love that. And yeah, we have been swabbing in our clean room quite a bit, actually, to understand how different construction of DNA labs, ancient DNA labs specifically, or low biomass labs influences their sort of background, but also to trace our background over time and space. So we'll have lots of swab stories. Maybe that needs to happen needs to get put into the title of that paper.
I think, and there needs to be a cartoon involved, of course.
Yeah.
But this is like really interesting to people. Now, what's going to happen? I mean, I've always interested in, quote, pure research, but I know for a fact that you've done some stuff that is applicable to oral health related to what you've learned from the paleo microbiomes.
Absolutely. So you're completely right in that we are learning a lot from Neanderthals and from other Paleolithic and Mesolithic humans and how we might improve our health today. So that was one of the most shocking things I learned as an early career researcher, was to go see Neanderthal teeth and Neanderthal jaws for the first time and see that they had amazing oral health and think, gosh, what happened? Why do we have such poor oral health today? Cavities are present in 90% of Americans. 90%. It makes it the most chronic disease in the United States. Moreover, periodontal disease is 50% of the American population that it hits, right? So oral disease are rampant now. They were not always in the past. And so as a young researcher, you're thinking like, gosh, did we just get this wrong? Like, what's going on here, right? And it was really this piece that we wanted to solve, or this puzzle that we wanted to go after. And so by understanding that the microbiome has changed several times since we shared microbes, or the majority of our microbes, with Neanderthals, by being able to track those changes and understand how the microbiome has changed, we now understand that we might be able to go back in time and really leverage the microbial ecology of past populations and try to use that to improve our health today, right? If we can go back in time and create these microbial communities that are, robust, for example, to acid production, or robust to many of the features that cause poor oral health today, we can really figure out how to translate this work into benefits for people. So absolutely. We started off doing ancient DNA and, understanding how these ancient human populations lived and how they changed their microbes over time and space. And we then leveraged that into looking at populations who live today. So we've worked with many different indigenous communities, and we understand how they may have unique microbes that are now associated with disease in several different ways. And we're able to sort of take all of that work and translate it into oral microbiome transplants. And so, like I said before, I now have a small startup where we're truly taking our science and translating it into a new tool, a new preventative technology for both caries and periodontal disease. Caries being cavities, and periodontal disease being maybe what you've heard of as gum disease. And we're really excited about this. We're in preclinical stages and we're moving into humans next year. But it's, for me, super personal, right? Because when you work with some indigenous communities and you see kids suffering from oral health problems, and they really shouldn't, and that's a kind of a colonial issue, right? Then we need to do something to change it. And, I also have somebody in my family who suffers from periodontal disease, and they are somebody that does all of the right things. They do all the things the doctor tells them to do, and yet they still suffer. And so if we can use this knowledge and use all the things that we've been learning over the last decade to improve people's health, like that's really what motivates me to get out of bed in the morning and do the work that we do. So it's not just, ancient DNA. It's not just, modern oral health research. It's really figuring out how we translate it and how we leverage it into new technologies tomorrow.
I think this is just amazing, and I'm especially moved by the fact that you are thinking about service to others with this, without making aspersions onto motives. When students that I talk to will see a commercial or an advertisement that eating a particular kind of yogurt could reduce depression, it really bothers me. And I understand that there was some work done on this, that, and the other. But to me, and I'm very old-fashioned about this, trying to be as clear and honest about the science is the most important thing to me. And being able to help others. I've said with great pride, there's nothing I've ever done research-wise that benefits a human being. So there. But those of you who do, I have a great deal of respect for. And it's so great to see people that have that strong opinion.
For me, it started early on when I was an undergrad at South Dakota State University. I actually have an undergraduate degree in sort of industrial microbiology. So I always thought I was going to use microbes to produce things that helped people. That was always my sort of wish. And so to kind of take this different path for a minute and study ancient humans and study, microbial communities in the context of health and disease, and to come back to that industrial microbiology degree and think about, gosh, how do we weaponize this in a way that improves people's health, right? How do we take this knowledge and actually make a difference in somebody's life? That for me became a really important goal of what we do and why we do. Why we do what we do. Because there's a lot of ancient DNA research and it's really interesting, but it's hard for me to stomach, just learning about somebody having brown hair and blue eyes and moving on to the next question. It's like, no, how do we take this information about the microbiome and use it to actually improve many of the chronic diseases that we experience today?
So you and I have both seen things in media suggesting that folks who lived, quote, in ancient times, end quote, had all kinds of health benefits. And I'm not going to address whether or not that's true or false. What I am going to say is that you are looking through your research of the molecular echoes of that period. And that's a lot different than what you want to be true. And I think there's a lot of us in society today that have very strong opinions of what we wish to be true. And the power here is that you actually see those echoes and can interpret them.
Absolutely. I had an awesome postdoc, Allison Ham, who's down at Grand Valley State, and we started really trying to dig through these unique microbiomes and microbiomes that existed in the past. And it's not just that we should say, oh yeah, everyone should have one of these ancient microbiomes because they didn't have as bad of oral health. And it's like, well, maybe they had worse other diseases, right, that we didn't know about. And so we've started doing a lot of paleopathological analysis, trying to understand some of these chronic or infectious diseases that people would have had and relate that back to their microbiomes. So, it's not always rainbows and, and easy stuff, right? It's really complex and it's really hard, but I think we can do the work. So I'm excited about that.
When I would teach introductory biology to people who didn't particularly want to be there, and I would talk about levels of genetic control, for example, and they would say, why are things so complicated? And I said, it's a good thing they are, because a lot can go wrong if you just have one switch. If you got a lot of switches, there's a lot of redundancy, and that's a good thing. And so whenever we look at what I'm going to say, which is essentially ecology, in the gut, in the teeth, on the skin, everywhere, we are students used to say that I don't remember saying this, that students are petri dishes with shoes. I don't recall saying that, but it sounds like me. What I would say is that we're actually a walking series of ecologies, and we really are. As you mentioned before, the microbes you find in the front of the mouth are different than the back. We know from a long time ago, right hand is different from the left hand. All of those things are true. And so how do you take that level of complexity and fish out stuff that's important? Makes it challenging, but rewarding.
Absolutely. And I think this ecological mindset is really where we need to head if we're trying to leverage microbiome sciences into improving health tomorrow. I always get people who say, well, why are you trying to transplant whole microbial communities when you could just transplant one or two microbes? And we could, but it's not changing the robusticity of what is an incredible community where there are these checks and balances and where there are these protections by, looking at many microbes together rather than just a single species or a single mechanism. So in my mind, this ecological mindset and how we think about these microbes working together and even how they work together from the mouth to the gut to the, everywhere else, and how these oral microbes can influence all sorts of things, right, elsewhere in the body. Thinking about this as an ecological problem rather than, an individual disease problem, I think is the way forward.
Oh, I agree completely. It's interesting. I'm sure you know this, but if you have a student who wishes to go to dental school or nursing school or veterinary, they're required to take microbiology, but not for medicine.
Yeah.
And I continue to say that that's going to change because all the problems we're talking about are ecological. And so this reductionism that comes very naturally to us because we've solved a lot of problems with the reductionist point of view. We really have. And then we teach students to look at, one gene, one phenotype, which of course isn't really true, but it's a simple way of looking at things. And this is really no different. There is no one magic microbe that will make you lean. There is no one magic microbe that will keep streptococcus from binding to your tooth pellicle. But there are groups of things that have this functionality. So we are a series of ecological niches.
Yes, and we should be leveraging that functional redundancy, right, in our microbiomes. If we know that there's many mechanisms, why are we not using them all at the same time, right? For me, it seems like a no-brainer.
No, I mean, when you look at, and a lot of folks are uncomfortable talking about fecal microbiome transplants, and especially when it comes to, and I always get the genus name wrong because they changed it again. It's not Clostridium difficile anymore. They changed the genus name to something else.
Yeah.
But the whole idea was you were transplanting an ecology. And I know people say, oh, we're going to find the core microbes that are necessary. And that's fine. But you'll notice it's not one magic bullet. That's the point.
Yeah. And there's a lot of amazing work, trying to find that individual, the single mechanism that will prevent C. diff. I think my concern is that it prevents it then, but then what is the community that comes back in? And is that robust again to a second C. diff infection? So for me, I want to think about like long-term robusticity to any new chronic disease that might come in rather than just a treatment again for that sort of single disease. So it might be that there's different tools that are useful in different scenarios too, and that it'd be best to have all of them at our at our disposal, right? But thinking about it both directions, I think is important.
I like that word robusticity because I think it's a good one. And it's really true because, I can slather on a microbe. And I think people have tried to do that, for example, for acne. No, there's a bacteriophage thing they're doing now. But the point is that, it's the community that's important. I don't know if listeners and viewers are aware of this, but if I swab the skin of a bunch of individuals, I will find the organism that causes MRSA there. It doesn't mean you have MRSA. It's part of a community. Mm-hmm. And it's complicated. Kind of like that's going to be the social media post. That's the Tinder thing. It's complicated relationship wise. They're down to conjugate, that kind of thing. The students make they come up with to make me laugh, but it really is true. It's a group of things working together, and it's true on your skin. It's true in your teeth. It's true on any surface.
Absolutely.
I don't know that I've told you this. We have found microbes in the water bottles that make antimicrobials. And I'm not surprised,
Yeah, absolutely.
So this is what's so exciting about the work you do. And you have a whole microbiome center at Penn State, too.
We do. We're so fortunate to have an amazing center here with over 500 different researchers, right? It's truly a powerhouse here at Penn State. It's one of the reasons I also came back here is just to be part of this microbiome community, because there's lots of people thinking about these ecological networks, how we employ them, how we can, collaborate with each other and feed off of one another and inspire each other, right? And so that just is a fantastic network to be part of, to think about these things, right? And to have also people challenge you on these things to say, hey, is that right? Or is that safe? Or is that okay, right? It's fantastic.
It's kind of like an ecology, isn't it?
Exactly. An ecology of microbiologists studying an ecology. Yeah.
Oh, it's true. I mean, you're at a small institution, you have a small number of voices, but now you have a lot of people working on the same area, not the same systems necessarily, and they cross-pollinate and fertilize. That's really exciting. I've noticed as we start to wind down a little bit that your original training was in industrial microbiology. What made you interested in microbiology to begin with?
Yeah, that's a great question. I was somebody who always wanted to be a medical doctor. And about the time I started my undergraduate degree, my grandfather passed away, and I spent a lot of time in the hospital. And I just thought, a lot of this is just helping people be comfortable. It's not actually curing the disease that ails these folks. And we need people who are working on new treatments and new items. And about the same time, some of the early microbiome research came out, and I was flabbergasted by the fact that there were all these microbes in our bodies doing all these things that we didn't understand, and just thought that was the most amazing thing ever. And so I finished my degree at South Dakota State and went on to Penn State and wanted to do microbiome research, and actually had an amazing time there doing my PhD in sort of biochemistry and microbiology, but there got to do infectious disease research and understand how microbes interact with the body, how they influence the immune system, et cetera. But I really realized for me personally that I wanted bigger questions. I wanted to be able to look across microbiomes over space and time and not just use mouse models. And so that's why I sought out ancient DNA and the ability to sort of create that into a model that we could leverage to reconstruct microbiomes over time and space. But for me, really being interested in microbiology stemmed from wanting to find those cures, wanting to find those reasons, and wanting to understand how these microbes and these diverse sets of microbes are involved in that. Because there's so much to learn. It's complicated, as we mentioned earlier, but I think it's the path forward. I think it's the way forward if we keep at it.
Well, as I say, we have to struggle. I'll give you a good example. When I see a change in an organism, the idea that that's due a bacterium, it's due to a particular genetic change, that's cool. But for $80 a sample now, I can compare that genome to the original genome and find out every gene that's changed. Some of them might not have anything to do with the change in phenotype that I'm looking at. But so the tools have become so much more powerful and so much more accessible. And that's the part that impresses me. I well remember when microbiome studies started, because I'm old, and it was interesting because people could say we can't analyze these things. Now, notice that you can, and it's getting better all the time. And as you say, if you're interested in the historical echoes of the different microbial communities associated with humans, since we don't have time machines yet, you found a way to do it anyway.
That's exactly it. We go back in time and reconstruct these microbes from people that lived in the past all over the world, right? We have the ability to literally jump in a time machine and go back and say, okay, these folks had these bacteria. And it sounds simple. It sounds like it's not much at all, but a lot of times you'll go back and see like these folks had all the microbes associated with gum disease, yet they don't have gum disease. What happened, right? And those I think are the sort of light bulb moments that we realize we can use this information in really positive ways for people today to bring that back from the time machine and employ it today.
Which brings me to kind of the final thing I'd like to ask you, if that's all right. Do you have like 2 or 3 things you'd like everyone to remember about this particular session?
Yeah, sounds great. I think the first one is that you should go out and try to challenge dogmas whenever you can. Don't just accept that something is the way that it is, right? We just accept that we have to brush our teeth and get rid of bacteria all the time. But in reality, Neanderthals had amazing teeth, right? We can not accept that as normal and understand that that's a disease state and we can go back in the past. So if you're a young student out there, I would say don't just accept dogmas, challenge them a lot. The second thing I would say is that science is always a learning process. Even now as a tenured professor, I am still learning every single day. You're never going to know it all. You're never going to get everything right, right? It's always iterative and it's always a learning process. And the last one is that to chase the things that you find passion in. That would be the last thing I would encourage students to really sit and think about. You can get up and do a job, but to do a job that you really are passionate about, I think is what really matters. I really care about getting some of this information from Neanderthals and from ancient humans into populations today in a way that can help them improve their health. And that means that it's easy to get out of bed in the morning. It's easy to do my job because I absolutely love and am, inspired by what we're doing. So find that nugget that you love. Don't just, chase around whatever your PI or whatever your professor or advisor is telling you to do. Think about what you want to do and what you love.
There's a famous observation people have made, and a couple of artists have made posters of me of it, and I'll try and put one I'll try to put one in here. But there's no feeling at all that can compare to being the first person to make a particular observation. I didn't say it was important in a cosmic sense, but no one saw it, or certainly said they saw it before you did. And that's what makes science so cool to me.
Awesome. I love that. Yeah, seeing a Neanderthal microbiome for the first time was one of those moments I won't ever forget.
Unbelievable. Well, you know what, Laura? It has been an absolutely wonderful session with you. I'm so excited that you could spend the time, especially because of some of the difficulties that I had on my computer, which folks don't need to know about. But I am so grateful for your time and the clarity with which you spoke today.
Thank you so much for having me, Mark. I really appreciate all the work you're doing to really get microbiology out there for everybody to understand and take home. I appreciate it. Thanks so much.
I think I'm wearing a microbial evangelist button, so that's what I'd like to be.
That's fantastic.
My very best wishes to you and yours, including your lab group.
Thanks so much, Mark.
You're welcome. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with, as always, epic links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Laura Weyrich can be found at Pennsylvania State University Anthropology Department and is lab director of the MicroArch program, as well as head of the Penn State Ancient Biomolecules Research Environment program. Many thanks, as always, to David Renata for superb editing and patience, and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.